Assalamualaikum warahmatullahi wabarakatuh and a very good day to everyone. In today's video, I am going to be presenting my final year project report with the title of Evaluation of Phytate Level in Soybeans by Different Wet Processing Methods. Before I start my presentation for today, I would like to introduce myself first. My name is Siti Hajar Aqilah binti Husain and I'm a final year student from UiTM Kuala Pilah. This will be the outline for my presentation for today. To start off, soybeans or soya beans, or is scientifically known as Glycine max, is a type of legume native to Eastern Asia related to peas, clover and crops. It has been used in a variety of diverse cuisines all over the world, which would include miso soup, doenjang, soy sauce, tempeh and many more. It has a high level of vitamins, minerals and proteins, and it also has a particularly high level of antioxidants and anti-nutrients. One of the anti-nutrients that can be found in soybean is phytic acid. So phytic acid is actually the primary phosphorus compound in seeds that would account for about 65 to 85% of the total phosphorus content in seed. In soybeans, this phytic acid fluctuated from 1 to 2.22%, and as it is negatively charged at physiological pH, it has an extraordinary chelating power with an affinity for various positively charged compounds in food such as minerals and trace elements. The problem statement in this study is that as phytic acid has an extraordinary chelating power for various positively charged compounds in food, it is well known for inhibiting mineral absorption. It binds to mineral and renders them inaccessible. Phytate cannot be absorbed or digested by the human digestive system or any non-ruminant animal if it is consumed due to a lack of endogenous enzyme phytase. When phytic acid is consumed, it will remain undigested, and when it is excreted, it will lead to a phosphorus pollution source. By doing this study, the more effective ways in reducing phytate levels will be identified and developed. Health issues or any mineral deficiencies that may arise in the future due to the level of phytate in the food can be reduced, and the nutritional quality of the food can be improved by degrading the phytate level in food. The primary objectives of this study is that to compare the effect of different wet processing methods on the level of phytate and magnesium content in soybean, and to study the effect of lactic acid bacteria that was present during the wet processing method on the level of phytate. These are the few topics and findings and the literature review that I have referred to throughout this research. The topics include phytic acid, phytate in soybean, magnesium in soybean, phytate reduction techniques, soaking, as well as accelerated fermentation. These are my research methodology. I started with collecting my samples from Econsif Kuala Pilah, and then I prepared the bacterial medium, which were MRS agar and nutrient broth, and I prepared the magnesium standard and phytate standard for UV-Vis and AAS analysis. And before any wet processing method, I start by detecting the phytate, magnesium pH and LAB count for the sample before wet processing. For the magnesium content in soybean, it was analyzed by using an acid digestion method. The phytate content was determined by using the Davis and Reid method. The pH was analyzed by using a pH meter, and the LAB was quantified on MRS agar by using a spread plate method. And here comes the first wet processing method, which was soaking. So during the soaking process, the sample was combined with sterile distilled water, incubated at 25 degrees Celsius for 24 hours. And after that, the soaked water was subjected for a pH determination. Subsequently, the lactic acid bacteria from the soaked water was isolated on MRS agar, incubated at 37 degrees Celsius for 48 hours, and then the LAB count was done. And after that, from the agar plate, a single colony was cultivated in nutrient broth at 37 degrees Celsius for 18 hours. This cultured lactic acid bacteria was combined with the sample alongside with the sterile distilled water in the process of accelerated fermentation, which was the second wet processing method. And after that, it was incubated at 25 degrees Celsius as well for 24 hours. LAB count was done for the sample after accelerated fermentation, and the pH determination was also done for the sample after accelerated fermentation. After both wet processing methods was done, the phytate and magnesium content in the soybean samples was detected, and the results obtained was analyzed by using one-way ANOVA, as noted by Noor, Haizan and Noor. For the results and discussion, it will include pH, phytate levels, magnesium content in soybean, as well as the LAB count. I will start with discussing the pH level in the soybean samples from before soaking, after soaking, and after accelerated fermentation. As can be seen from the slide, the pH levels in the sample from before soaking, after soaking, and after accelerated fermentation shows a significant difference from before and after each wet processing method. The water used also became more and more acidic. Before soaking, the sample's pH was 6.30. After soaking, it is 5.31. The pH drops because during the soaking process, the lactic acid bacteria begin to grow due to the natural fermentation, which is known as lactic acid fermentation. So during this process, the ammonia and lactate was converted into lactic acid that causes the pH to drop. From after soaking to after accelerated fermentation, the pH drops even more from 5.31 to 4.96. This is because during the accelerated fermentation process, the sample was combined with isolated lactic acid bacteria. So this lactic acid bacteria will undertake the lactic acid fermentation of the sample and will form lactic acid that will result in the reduction in pH. Hence why from after soaking it is 5.31 and after fermentation it is 4.96. Next is the phytate level in soybean. From the left side of the slide, a calibration curve for phytate standard is shown, and as can be seen from the table, the phytate levels also shows a significant difference from before and after each wet processing methods. Just like pH, the level of phytate also decreases after each wet processing method. From before soaking, it is 0.36. After soaking, it is 0.21. This is due to the release of lactic acid during the fermentation process that leads to the reduction in pH. This will then promote the increased activity of the endogenous enzyme phytase. Endogenous enzyme phytase is actually the phytase from the insides of the soybean, which means that it is a plant-based phytase. As noted by Zoo 2019, this phytase is a phosphatase subgroup and it will help in the dephosphorylation of the phytate. From after soaking to after accelerated fermentation, the phytate levels in the soybean sample degrades even more from 0.21 to 0.095. This is because, as mentioned previously, during the process of accelerated fermentation, the sample was combined with the cultured lactic acid bacteria. So this cultured lactic acid bacteria, as noted by Kumar and Sinha, bacteria such as E. coli, Bacillus, Lactobacillus and many more can also contribute to phytate degradation by producing phytases, which is known as extracellular phytase or microbial phytase. This microbial phytase, together with the endogenous enzyme phytase, works together and degrades the phytate levels in the soybean sample during the process of accelerated fermentation even more, and hence decreasing the phytate levels from 0.21 to 0.095 in the sample after accelerated fermentation. Moving on to the magnesium levels in soybean, it can be seen that as compared to pH and phytate levels in soybean, these magnesium levels increases after each wet processing method, and it also shows a significant difference before and after each wet processing method. From before soaking, the magnesium levels in soybean was 0.095 milligram per grams, and after soaking, it increases to 0.15, and after accelerated fermentation, it increases even more to 0.23 milligram per gram. During the process of soaking, the magnesium levels in soybean increases due to the activity of the endogenous enzyme phytases, which is plant-based phytases that helps to degrade the phytate level in soybean samples. And when the phytate levels is degraded, the minerals, which is magnesium, is no longer bound to the phytate, and it is released during the degradation of phytate in the soybean samples after the soaking process. From after soaking to after accelerated fermentation, the magnesium levels in soybean increases even more from 0.15 to 0.23 milligrams per gram. During the accelerated fermentation process, the sample was combined with the cultured lactic acid bacteria. So the cultured lactic acid bacteria provides a microbial phytases that works together with the endogenous enzyme phytases, degrades the phytate even more, and more minerals, which is magnesium, are released, hence increasing the minerals, which is magnesium, after accelerated fermentation. Last but not least, I am going to be discussing the LAB count for the sample from before soaking, after soaking, and after accelerated fermentation. Before soaking, the LAB count was 0, meaning that there are no lactic acid bacteria that was detected on the MRS agar. After soaking, it increases to 114, and after accelerated fermentation, it increases to 156. Please note that during the soaking process, the water that was suspended on the MRS agar was diluted 5 times, while the water that was suspended on the MRS agar for the after accelerated fermentation process was diluted for 6 times. The serial dilution is necessarily done in order to obtain a desired colony growth within an acceptable range of 30 to 300 colonies. The colony forming unit of the bacterial count can be obtained by using the formula C times M over V. The colony forming unit can be identified by using the formula C times M over V. C is the number of colonies, M is the dilution factor, and V is the volume of culture that was suspended on the MRS agar, which is 0.1 ml or 100 microliter. These are the colonies of LAB on MRS agar from before soaking, after accelerated fermentation, positive culture and negative culture that acts as a positive and negative control in this process. To summarize everything that I have discussed earlier, the effect of different wet processing methods, which were soaking and accelerated fermentation, on the level of phytate and magnesium in soybean were successfully identified. To significantly lower the phytate levels in soybean, the process of accelerated fermentation is preferable. This is because during the soaking process, the phytate level degrades, but the phytate level degrades even more during the process of accelerated fermentation. And during the soaking process, the magnesium levels increases, but it increases even more after the process of accelerated fermentation. This is due to the presence of lactic acid bacteria that was combined with the soybean sample during the process of accelerated fermentation that helps to accelerate the degradation of phytate in soybean. In future studies, various approaches could also be used to enhance this degradation of phytate in sample during food processing. For example, by giving a more favorable conditions for the phytase activity, by adding more isolated phytases from plants and animals, or by using raw materials with a high intrinsic phytate degrading activity. These are all the references that was used. That's all from me. Thank you. Don't forget to give this video a thumbs up if you like the way I explain my research project report. Bye bye.